All right, so we'll go ahead and get started so that Dr. Uribe can have all the time that she needs. So today I have the pleasure of introducing to you guys Dr. Rosa Uribe. She's an assistant professor of biosciences at Rice University, as well as a CPRIT scholar in cancer research. Dr. Uribe earned her PhD at the University of Texas at Austin, and then went on to do a postdoctoral fellowship at California Institute of Technology, where she primarily worked with zebrafish to understand early embryonic development using invertebrate organisms. Today, Dr. Uribe is going to be our second speaker in our series of innovation. Our second speaker in innovation and diversity in biomedical sciences. So the primary purpose of her talk today is going to be directed at graduate students or predoctoral students, as well as postdoctoral fellows, to enable them to better understand the journey of underrepresented faculty and how they have gone through the world of science. Speakers will include underrepresented professionals in biomedical research and research-related careers. So I'm really excited to hear about how Dr. Uribe got to where she is today. So I'll go ahead and hand the talk over to her.
Thank you so much, Joe. Thank you to the Keck seminar series, and thank you to GCC for enabling a platform for me to talk to you all today about my journey in science and some of the recent work that we've been doing in the Uribe lab since I've arrived at Rice University. So I'm going to share my screen and I'll go ahead and get started. So today I titled my talk, Seeing is Believing, Unraveling the Mysteries of Embryonic Development, One Experiment at a Time, because you'll see as I describe my journey, it really truly has been all about embryonic development, whether or not using more biochemical methods or cellular biology methods, or all the way up to the systems whole embryo level. I feel like I've flipped every rock and stone, and we're still trying to understand how specific cell types develop in the vertebrate embryo. But before I talk about the vertebrate embryo, I just really wanted to take a step back and tell you all a little bit about me and some of my early entrance into science. So as the title says here, I didn't really know that science was done at universities. So when I was growing up as a child and even as a teenager in San Francisco, I was born and raised in the San Francisco Bay Area. Science was just really like an abstract thing. I knew I was interested in it, and I knew that I was interested in biology, but as I've written here, just to really make it clear, it was not anything that was made obvious to me in media, TV, cartoons, whatever, any kind of, disseminated source that being a scientist was for someone like me. So medical school was the main option that was communicated to me over and over again, like not only through school and high school, but just in the media. Also, the second point that I have here is that even when I did see, scientists in the news or heard about them or read about them, I never saw anybody who actually did science in real life. So growing up, I come from a Mexican-American family, and it's just a cultural thing that we work for a living, and my family was a working class family. And so my whole family was just, that's what we did. We lived pretty much paycheck to paycheck, and life was just about making sure that you're comfortable enough to get through the day. And the idea of doing science, let alone, from anybody who even looked like me, was not even a thought or a concept. And so I did really well in high school. I went to Catholic high school, and then I decided to go to college with in mind pre-med, which is really common for people like me where I grew up. So I started at San Francisco State University in the heart of San Francisco, and was taking just basic biology classes and the typical pre-med, the typical pre-med track here. So I was going through freshman year and sophomore year, and it was at the end of sophomore year. Before I got to sophomore year, I was having some problems, and it ended up being that, like, you know, coming from a working class family and my background, it was very difficult to focus on my studies. And I was working many jobs in addition to I had a full-time retail job while I was trying to take biochem and organic chemistry or whatever and all that. And it was very difficult for me, not because I didn't understand it. It was just, I just didn't have time. So everything changed for me when I decided to just quit all my jobs and take out a whole bunch of student loans and focus on the science. And so when I ended up taking an upper level genetics class, my whole world changed. Walk into the classroom and Dr. Ramirez Weber, Felipe Andreas Ramirez Weber, was the instructor. He was the professor teaching the class. And my entire time being at State, all of the professors, almost all of the professors that I can remember, or even in high school, were people who did not look like Dr. Ramirez Weber. He is no longer with us today, and I'm going to tell you a little bit more about him and his inspiration for me. But what happened was he was enthralling. He was teaching genetics. He was passionate about it. He had recently finished a postdoc at University of California, San Francisco. And what was he studying? So he pioneered studies on, he discovered a new way that cells within an epithelial context. In his context, it was the Drosophila wing disc. So basically, before a fruit fly becomes a fruit fly, it goes through a maggot, what we commonly know as maggot phase. And then the maggot undergoes basically a metamorphosis, and then it transforms into a fruit fly. When it's still in that maggot phase, the cells are talking to each other within the epithelial sheet and turning into, helping each other to pattern into, am I going to become a wing cell or am I going to become a leg cell? And so he discovered at UCSF a revolutionary new way that cells communicate via these things called cytonemes. And I won't go too much into the cytoneme story. I actually have a very elaborate Twitter thread on this if you're interested in actually checking out my Twitter page. It's just basically at Uribe Lab Rice. This is the Twitter story, but basically, to make a really long story short, he discovered that there are these really long cytoplasmic, thin filipodial-like extensions that will either carry ligand to from a signaling source to a receiving cell, or the converse. They will emanate to a signaling source, and they will have receptors on them, or a combination. And so he sat down and drew this all out for me, young Rosa, sophomore Rosa, and I decided that I just really wanted to join his lab. I didn't know that undergrads did research at a university. I didn't know that I could do that. It was just something that I was frightened of. And I remember just being really, really, really scared and asking him, do you even let undergrads observe? Like, I didn't even know it was a thing. And he let me in. And I ended up learning a lot in that lab, being immersed in hardcore Drosophila technique right off the bat. I started off learning how to wash dishes and make fly food, and doing larval dissections to dissect out imaginal discs, and doing a lot of molecular cloning in order to generate some biochemically altered candidate ligands to understand if they go from here to here, like to visualize them in the wing disc. So I ended up graduating SF State with a really strong sense of understanding about developmental biology and how cells might signal to each other. But when I was at SF State, and after I joined the Ramirez Weber lab, it was specifically this program, this phenomenal program that many of you might have heard about it. Many of you might not have heard about it. At the time when I was an undergrad, this was a novel thing. Like San Francisco State was one of the only universities in the entire nation. I think there was just a handful of pilot schools doing this, where the NIH provided basically a training grant to these undergraduate institutions, primarily minority serving. And they ran a training program for undergrads that would enable them to join a lab in their junior and senior year and pay. It was a full scholarship, so it would pay for your tuition and fees, so you don't have to worry about anything, but it would also importantly provide you with a stipend. And so for me, that was transformational. I didn't have to worry about working a retail job. I was able to just take a stipend, kind of like a mini version of a grad student. But even then, it was meager funds, but to me, it was like the gold and all the riches in the world, because it enabled me to focus on my classes, but also go to the fly lab and do my fly studies without worrying about waking up and doing the night shift. For example, at Nordstrom, where I had been working before, dealing with rude customers. And so Dr. Frank Bayless and Dr. Raymond Isquer, who's now the current program director, Dr. Frank Bayless was phenomenal. He brought me into his office, set me up, and basically said, you should apply to this. You're a perfect candidate. Now that it looks like you've been doing some research with Dr. Ramirez-Weber. And I was super naive. I was like, okay, whatever, like I'll apply to this. They're telling me to apply to it. And I didn't fully understand what it was. I don't think anybody told me that it was a $20,000 scholarship. So I remember just filling out some forms that they gave me. Back then it was where they actually gave you a form and you wrote things out in pen. Like that's how the application was. And then they were just asking for some transcripts. So I applied and then I got it, and I ended up getting the scholarship, and I decided to spend a whole extra year in undergrad to just fully ride out the scholarship and finish my studies in the Ramirez-Weber lab. In the end, I obtained just to go back a slide, wonderful experience at SF State. And while I was applying to graduate school, after he told me everything that I needed to do, and he insisted that I leave California. I wanted to apply to all the UCs, Stanford, all that. He said, yeah, like I'll write you a letter of rec for those places, but I insist that you apply to some out-of-state schools, because I'm not going to write you a letter of recommendation unless you do that. And he was just a phenomenal mentor, and it meant a lot to me. So I did, and then I ended up, to make a really long story short, I ended up going to UT Austin after that. So I got into UT Austin, and again, super naive, didn't really understand how big of a school it was. I didn't know until I actually got there. I got accepted to UT Austin into back then, well, actually, I think it still is known as this now. It's the Institute for Cellular and Molecular Biology. It was a big umbrella program at the time. I was one of 35 entering graduate students who were either interested in developmental biology, structural biology, biochemistry, the whole gamut at that graduate program. And I ended up joining the lab of Dr. Jeffrey Gross, who was just from the beginning one of the most kind people I've ever met in my life. And he took me under his wing and showed me how to use zebrafish as a model organism. I'm going to tell you a little bit more about that if I have enough time after telling you all about myself. I'll tell you about the zebrafish model that we're using. But he took me in the lab. I was he was a brand new PI at the time. He had only been at UT for about a year or a year and a half when I joined. But essentially, I learned a lot. I was immersed in a rich culture at UT Austin, a really rich culture of postdocs, faculty, and a huge graduate student population. And when I say rich culture, I mean everybody was super collegial. Everybody knew each other. Of course, the graduate students hung out and we talked amongst ourselves, but there was this unique thing that I have yet to really see since I've left there, that the faculty in the Institute for Cellular Molecular Biology and some of the new assistant professors who were there at the time, besides Jeff Gross, including Steve Vokes, Johann Eberhardt, and John Wallingford, they basically just treated us all like colleagues. It wasn't like, oh, you're just a graduate student. Yes, it was like, yes, I'm your advisor and you're working for me and we're learning together, but at the same time, it was collegial. I felt truly included and that I was seen as an equal. And so this was across all spectrums. And one piece of advice that I like to give graduate students and even postdocs who might be starting off or are part of a network is that please don't be afraid to reach out to faculty and just talk to them, talk to your committee, just, hey, how was your day? Oh, I went to this concert the other day. You know, just talk to them and you'll see that everybody's a human. We're all humans and we genuinely care about you and your life and we want to know about you. And we would all go out to the pub together. It was quite regular, in fact, that us students would go out with postdocs and faculty, and we would just all march down to the pub, the Crown and Anchor, and just and just really talk science for hours and hours and hours over beer. It was like such a great experience. So while I was in the Gross Lab, I learned a lot, both off the bench as well as on the bench. And he really enabled me to truly develop my developmental biology skill set, as well as help me to learn how to even just think like a scientist fully, the whole experience. And so I'm just showing you like a representative image from one of the main papers from my graduate work. Essentially what you see here are zebrafish eyes that have been sectioned through, and all of the green signal are basically the nuclei. So that would be similar to DAPI, but this is cytox for those who might know. And you can just see two specific retinal neuron markers shown in a control situation, or in this one candidate gene that I happened to be looking at throughout my graduate work, where you can see that there's a clear phenotype. Compared to control, the eye is smaller. And histologically, you can see that the beautiful layers that you would see here in the control are really pathetic looking for multiple different cell markers that were looked at. And in the end, what I ended up discovering is that compared to just a normal retina that I schematized here in a cartoon that would express these genes within these particular developing layers. So for example, this one gene, ATH5, would be expressed in this inner layer and largely responsible for the formation of this one cell type called the ganglion cell within the retina, so on and so forth. And we saw that this was just basically quite perturbed when the fish no longer had this one protein called id2A. So what I ended up following up on with this, and this is just a snapshot of 4 papers out of a series of papers I ended up doing out of the lab from my graduate work under the excellent mentorship of Jeff, and just being enthralled by the science and just really grabbing my project by the horns and really taking full ownership of my project. And I'm not going to go into all of these studies. I just wanted to highlight the fact that I was able to, get this body of work out that really put this one factor on the zebrafish retinal differentiation scene. And since this work has come out, there has been more appreciation for the specific, this type of gene. It's called inhibitor of differentiation, how these inhibitor of differentiation programs, there's 1 through 4, id2 is just one, the second one of the 4, how they're these bizarre proteins that are kind of shuttling in and out of between the cytoplasm and the nucleus, depending on what they're bound to. They're not transcription factors, but they're also not necessarily scaffold proteins. They're kind of just these like dominant negative pieces of protein that are floating around, and they bind things and sequester things into different compartments to influence retinal differentiation. So that was interesting. And then I did some follow-up work to find that it somehow limited notch signaling pathway output in the retina in order to modulate retinal development. But after that, what I ended up doing is I decided to stay in zebrafish and just move on and fully immerse myself in a new question for postdoc. And continuing with this theme of community and inclusivity, I joined the Bronner Lab at California Institute of Technology. And this was truly something that I didn't think that I was good enough for, to be honest. Even coming from this department at UT Austin, I didn't think that Dr. Bronner would even look at my application. And this is my piece of advice to graduate students. Don't ever think you're not good enough. You are. Jeff had to basically grab me by the shoulders and shake some sense into me about, oh, you should really do the science that you want to do and don't be afraid or intimidated by the stature of a lab. And Marianne Bronner is a giant in the field, and she's really known for pioneering studies on neural crest cells, which I'll tell you about here in just a few minutes, where basically her main question as a cellular and developmental biologist is, how do these stem progenitor cells known as the neural crest, how do they actually turn into different cell types in the embryo? So for example, how does it know to become a neuron or glial cell versus a cartilage or a bone cell? And so I joined the lab in 2012, and this is a picture of 2015, well into the postdoc. But this is even just a portion of her lab. A lot of these people have gone on to either head their own labs now, or they are about to, and they're spread all around the world. And this huge lab and this sense of community allowed us to support each other scientifically and fully immerse ourselves intellectually into the science. And so it was in this environment that I knew this was a huge field. It had been around for like 100 years. What else could I possibly discover about neural crest cells and how they know to become a neuron versus a cartilage cell? What could I possibly do? And so that's when I decided that we didn't know much about this derivative that I'm going to tell you about called the enteric nervous system. It's a derivative of the neural crest. And so with that, I ended up taking my research ideas and applying for a faculty position here at Rice University in the Biosciences department. And as you can see, now we're running a pretty big lab 3 years later. And I arrived at the end of 2017, but we really got our fish facility built and everything fully up and running in early 2018. And I'm just so amazed at how much we've done even since then, and despite the pandemic. And these are just some snapshots of some of the pretty pictures that we've obtained from the lab so far. But essentially we study both, we're at the intersection of studying both just pure neural crest, as well as all the different progenitor cells that are going to be budding off of the neural crest lineage and differentiating into enteric nervous system during development. Okay, so now I'm super excited to tell you all about a little bit of the science. So with that, I'm going to tell you about, I can't tell you about everything, right? I'm going to tell you about the most recent story that we've put out from my new lab at Rice University, looking at this intersection between neural crest stem cells and enteric nervous system. And I'll define what all of this is in the talk here. Okay, so what are neural crest cells? Neural crest cells are a special type of stem cell. And they exist so far as we have seen and detected in all vertebrates to date. Some might argue that in an invertebrate, some neural crest-like cell type might exist, but that remains controversial. But basically, in very early embryonic development, when we are largely still a flat epithelial series of epithelial sheets, there's one region that has started to turn on neuronal bias in the early embryo. And basically over time, as that epithelial sheet begins to fold up into a tube, and this tube is called the neural tube, and it is the precursor to the entire central nervous system, including the brain and the spinal cord. It'll it'll roll up from a sheet, from a sheet to a tube, and there are already some tissue domains that have been specified along this tube. The neural crest are specified. I'm not going to go into some specifics here, but basically what you need to know is these cells exist within the neuroepithelium. They're epithelial. As soon as the tube forms, they undergo an epithelial to mesenchymal transition, which means that they will transform from an epithelial cell to a mesenchymal cell. They break their attachments and they will crawl away from the neural tube. They will migrate away from the neural tube. And what my lab studies is, after those cells have delaminated from the neural tube, how do they know to become all the different cell types that we know today that neural crest cells will give rise to? So for example, we know that neural crest cells will give rise to everything from the melanocytes, or the pigment cells within our skin, all the way to neurons and glial cells or smooth muscle cell. And so with that, I ask these two main questions with this recent work in the lab. Specifically, what are the neural crest cell terminal differentiation programs? Like, what are some of those gene circuits that are activated to become melanocyte versus neuron? And while they're going from a stem cell state to that differentiated cell state, what are some of the progenitor cell states that exist between that? So there's a lot of stuff happening between point A and point B. And we know already from a lot of previous research in other vertebrate models, such as mouse, chicken, as well as Xenopus the frog, that there are a core series of signaling molecules that are expressed along that neural field, such as Wnt's, BMPs, FGFs, and Notch signaling, which will impinge upon the activation of these master regulators of neural plate border identity. This neural plate border is basically the red region that I was just showing you in the previous slide, such as the ZIC and the MSX factors. There will be a true bona fide neural crest specification module that is very conserved across all vertebrates to date, largely, that have been examined, including FOXD3, the transcription factor SOX10, and the snail transcription factors, which will allow them to undergo EMT and migrate away from the neural tube. As they're migrating, there are certain genes that are required during their migration, and we'll go into that more here. And we do know some details about, as they're migrating to different destinations, some of the gene regulatory modules that are activated as they're becoming a neuron cell versus, for example, a pigment cell. And we know that zebrafish is coming more and more relevant in this field, highlighted by the fact that there was a recent article, a review article in 2020 summarizing the state of the field, that we have large conservation between each of these steps. You can see a lot of the similar players leading down to what we know about these derivatives. And a really obvious factor that comes across these vertebrates is known as SOX10, and you can see that it's expressed at multiple levels of the gene regulatory network. And zebrafish are fantastic for studying this, because not only are they rapidly developed, so you can go from a fertilized embryo, to an embryo that has a primitive eye and a trunk by 24 hours post-fertilization. They develop external from the mom. You can see it's in its chorion, and then it's transparent, and it bursts out of this chorion protective layer a day later, and it can swim and eat, and it's still largely transparent by the 4th day in development. And we have lots of different transgenic lines that will label with fluorescent proteins what we're looking at. And so as an example of that, graduate student Adam Howard in the lab has been using a lot of live imaging to understand just these early migratory patterns of the SOX10 positive cells, which are basically the marker for the neural crest cells. So when we take our confocal microscope and he focuses it here on this region of the developing fish embryo, you can see that the neural crest cells labeled here with the SOX10 are already really abundant from the head to the tail, or the anterior to the posterior of the embryo. But if he takes a time lapse between the 25th and the 47th hour post-fertilization, what I hope you can see right away is that these cells are highly migratory during this stage. So this is after they've undergone EMT and as they're migrating to those tissue destinations to become either the pigment cells, the neurons, or the glia. So we were curious, what are the transcriptional programs that these cells, right at these stages, what are they expressing? But before I tell you about that, I need to zoom in and tell you a little bit more about what else we knew about this. So we know that depending on if the neural crest cell is born, because basically as the neural tube folds up, it's happening along the entire anterior to posterior length of the fish. So you have a future head and future tail. Depending on if it's born in the head, the trunk, or this middle region called the vagal neural crest region, they have been known to become specific cell types. The ones that will eventually go on to form the enteric neurons are largely from this middle segment of neural crest cells that are born right here in the future neck of the embryo. Okay, so I'm going to finally tell you what the enteric nervous system is. So what the heck is the enteric nervous system? Okay, so the enteric nervous system are a series of hundreds of millions, in humans, hundreds of millions of neurons and glial cells that together are interconnected and span the entire GI tract. So basically from the esophagus all the way to the anus. And in a cartoon form, it looks very simplified like this, that these neurons and glia are bundled together in these ganglia, which are all interconnected with each other in the inner layer. And then there's a large outer layer as well of ENS. And these are all intertwined between both the circular and the longitudinal muscles of the entire gut tube. They help to regulate gut motility or peristalsis, so movement of food through the gut. And they also help with feelings of hunger and fullness and secretion of digestive hormones. Now, there's bidirectional communication between these nerves and extrinsic nerves that run all along the spinal cord and up through the parasympathetic and the sympathetic innervation, as well as the vagal nerve. And so it's really important. You kind of need an enteric nervous system in order to live. So we're interested in how it forms and from the neural crest cells. We know that in a mouse embryo, for example, that it is these vagal level neural crest cells. So the neural crest cells that are born at this junction of the embryo that undergo EMT, there's some reason why they know to migrate into the primitive gut tissues. And they will undergo this spectacular migratory event where they will reach the distal end of the gut tube. We know that from some other work that SOX10, the transcription factor SOX10 is important for this hierarchy in order to activate downstream factors that are required for the migration of these cells down the gut. And then eventually with some other transcription factors, they will turn on neuron subtype gene programs such as inhibitory neurons or excitatory neurons that will eventually form in the enteric nervous system. In zebrafish, how does this happen? So we could use the same SOX10 line that I just showed you before, but this one uses membrane RFP in order to label the cells. So this time, instead of showing a shot from this region of the embryo, we took the fish and looked right on its side and zoomed into its little developing gut tube right in the second day of development. And what you see here is this, and I've outlined the gut tube for you here with these dashed lines. And what you can see right away, before I even play the video, is that there are a whole bunch of cells that are still SOX10 membrane RFE positive, but we have some of these cells that are migrating in these streams along the gut tube from the anterior to the posterior, or from another way to put it is from the mouth to the anus side. And so this is also spectacular. So this is happening dynamically as the larval fish is itself growing in size at the same time that it's learning how to swim. So there's all kinds of stuff going on. What are these neural crest cells expressing as they navigate the gut environment? What are these other neural crest cells doing that we see above the gut tube? What are they expressing? And I've told you a lot of information here that these neural crest cells go from a stem cell state to a neuron really quickly. So roughly between 32 hours and 96 hours post-fertilization. And so that's by the 4th day in development. And they're doing this while they're navigating the growing gut tube of the larval zebrafish. And so we went and took a step back and returned back to the SOX10 GFP line, because we know that in the 2nd day in development, which is right at these steps right here, that not only is it labeling those cells as they're migrating along the gut, but all these spinal cord cells, these neural crest cells that have streamed into the developing phase. What are all of these things? What are they expressing when they differentiate? So we took a single cell approach to dissect this. And I just really don't have time to get into all the specific details, but what I can tell you is we took 2 different developmental stages. We dissected these axial segments from 100 embryos at this time point and 100 larvae at this time point. I got a little tongue-tied there because I wanted to say that between this time point and this time point, they technically transitioned from being known as an embryo to a larval fish. So that's like a little bit some specifics there. We basically used flow cytometry to isolate all of these cells from that dissociated tissue and inputted it into a 10x genomics platform. And we ended up capturing thousands of cells for each main time point. And so what did we find? So if you look at both of the time points, I didn't know what to expect. Us as a lab, this was a lab collaborative thing. We weren't quite sure. I had some bets about how many different cellular populations we would find, but essentially when we used the computational analysis in order to understand what these cells were expressing, we found, using this program called Seurat, 42 distinct cellular populations. And I'll tell you what that means here in a minute. So at the 48-hour time point or the 68-hour time point, this heat map here illustrates to you that indeed we have either 0 through 18 or 0 through 22 transcriptionally distinct cellular populations that were detected from all of those SOX10 GFP positive cells that we had isolated. And the heat map beautifully illustrates that by these yellow enrichment bars are the most highly expressed genes. So these are the gene signatures for each of those distinct populations. And the t-SNE further drives that home and color codes it for you, where you can see 19 clusters at this time point, 23 at this time point. And this is just a graphical way of displaying, an alternative way of displaying the data. So you can see the relationship, for example, between cluster 0 and cluster 18 based on color. And to summarize, we wanted to understand, out of all of those cellular populations, there were 42 of them, what are they? So we ended up classifying those 42 populations into 8 major cell type categories. And they're summarized here again in heat map, where we found, and as well as t-SNE color coded, where we found that cells existed with what we're calling a mesenchyme identity, whereas on the other end of the spectrum, we detected cells that were expressing transcripts that denoted either neuronal identity, neural identity, glial cell identity, or pigment identity, for example, between the 2 time points. And that's schematized here in a cartoon form based on where we speculate these cells would be found in the embryo or the larval fish at this time. But one striking difference between these 2 time points, they look very similar, was the fact that at the earlier time point, we found cells that still had a neural crest stem cell signature. So they were expressing some of those gene regulatory neural crest specification markers that I had introduced to you in the beginning of the talk. And this was very interesting to find. And it was something that reviewers also found very interesting. And I say that because we know that at 48 hours post-fertilization, that neural crest cells have already undergone EMT, and they've already delaminated from the neural tube a long time ago. Like we're talking 30 hours ago, or at least over 24 hours ago. So cells that were retaining the signature suggest that there's a core multipotent neural crest stem cell pool that still exists at these later stages. So what we then did is we put all the major cell type markers into categories. And so what I'm showing you here is based on the cell type that we had annotated. I'm showing you the major cell, the major genes that denote each of these. So for example, muscle cells, which you can see are highly enriched for these 4 genes here, are expressing these well-known muscle markers, whereas on the other hand, cells that were expressing genes that we knew were supposed to be enriched in neurons, such as ELAV-like-3, ELAV-like-4, they were strongly enriched in the cells that we were classifying with a neuronal identity. And specifically the neural crest cells, again, here's proof that we found some of these enriched factors. So we knew that SOX10 transcript itself was enriched, as well as some other specific neural crest markers. So that was really interesting to us. And so what graduate students Joshua Moore and Philip Baker did, in addition to with a lot of input from the rest of us, but they really largely led this part of the work, was they rolled their sleeves up and took a closer look at those cells within the population. And so not only when looking at the categories again, not only when they looked at this panel of neural crest transcript markers, not only did they find the SOX10 and the other neural crest markers that I had previously mentioned, but some other ones when they went back to the literature that were described to be expressed in these cells, as well as cells that were expressing known markers for an enteric fate, such as FOX2B and RET. And we knew that a recent review, a very recent review in 2021 had been done, just really summarizing some of the major markers for the zebrafish enteric nervous system field. And you can see that we found strong enrichment, for example, for RET or FOX2B or MEIS3 shown here. Okay, so I want to keep an eye on the time, and it looks like we have 42 after. I'm just going to go for a few, just probably for about another 5 minutes to make sure we have enough time for questions. So I'll just keep going. And if you have any questions, either put them in the chat, or we could also unmute and talk at the end. But just to keep going and to kind of get through this part of the story, just to back up for a second, so we found these markers. So that was great. It showed to us that indeed within those SOX10 GFP positive cells that we had sorted and submitted for single cell RNA-seq, we found that not only were some of these cells expressing bona fide neural crest stem cell markers, but they were also expressing cells that were slightly biasing them toward an enteric nervous system fate. And so what we wanted to know is, okay, so well, that was our first time point. Do we actually capture cells that are starting to turn on neuron programs, enteric neuron programs a little bit later? So what Philip did was he confirmed that we can actually see this in the embryo first. And so I forgot about this for a second, but basically just to tell you about this, what Philip ended up doing was he used a known hybridization chain reaction to understand back in the embryo where FOXD3, Crestin, GFRA, and NGFRB, these transcripts shown here, where do they localize in the embryo? And it's a multiplexed, basically it's a fluorescent in situ hybridization. And so when he zoomed in here to the developing gut tube of the embryo, he indeed saw that there were cells co-expressing these factors all at the same time. So that was great. We confirmed that they were expressing these markers, but like I was saying, we also wanted to know, okay, so we have these cells, what are they expressing right when they're transforming into neurons? Can we detect that? And so what Philip did is he went and looked at the 68-hour time point, and he looked at a panel of markers, and instead of by category, I'm showing you the raw cell population. So populations 0 through 22. And what he found right away when he looked at a panel of enteric markers as well as neuron markers and markers that would denote a more differentiated neuron subtype, strikingly, cluster 12 and cluster 5 were really strongly enriched for this panel of markers. And so when he went back to the embryo and used the HCR method to simultaneously look at ELAV-like-3 and or FOX2B, NOS1, and VIP, which again is a nice spread of these differentiation markers, and I'll remind you where they exist in this enteric neuron hierarchy. They exist here. He was able to confirm that along the zebrafish gut, not only are cells expressing this enteric differentiation marker with an inhibitory marker, NOS1, but also this early neuronal marker, ELAV-like-3, with another inhibitory peptide, VIPB. And so Philip was fascinated by this and wanted to go further. So what he did was he subclustered out populations 5 and 12 and reran an analysis. And what he ended up determining from those cells was that 5 new clusters were born, which we're calling the enteric neuron subclusters. And so to show you what this looks like in a dot plot, we indeed saw that the brand new enteric subclusters 0 through 4 were all expressing the enteric markers FOX2B and RET. But right away, he noticed that the one subcluster that was expressing the more differentiated markers, VIPB and NOS1, were enriched in subcluster 3. And so when he went and analyzed this further, he saw that not only were they expressing these inhibitory neuronal markers, but he knew from the literature that maybe these might mean that these cells are starting to turn on a sensory neuron signature. And so when he queried for these sensory neuron signature genes, he indeed found indicators of this one particular subtype called the IPAN emerging, denoted by these transcripts here. And he confirmed this in the embryo by performing a 5-color hybridization multiplexed hybridization chain reaction back in the embryo. And that's shown here very nicely along the gut. And so I'm going to skip over this because I just really want to just cut to the chase, that he found further investigating that we found transcripts that suggest that the opioid pathway is active in that developing sensory neuron signature. And that we have found some new information using a combination of single cell biology and the zebrafish embryo, that these enteric progenitor cells are very dynamic, they're migrating. And we have found the first known signature in zebrafish showing that, that the opioid pathway might be playing a role in this process. And with that, I wanted to just thank the lab. Everybody played a role in this last study that I just told you about. And I really just cut to the chase. There was a lot more data that was attached to this study. We have a recent eLife paper if you wanted to find out all the interesting information and other things that we found out about the study. And I just really want to especially thank co-first authors, Adam Howard and Philip Baker, for their stupendous work along with the rest of the single cell team in the lab. And I'm going to end because I've been yapping for a really long time, and I'm really hoping that maybe you guys might have some questions about either me, my journey, a little bit about the science. Anything is fair game here, and I'm happy to answer any questions.
Dr. Uribe, thank you so much. That was so inspiring. I have questions about your story, but I wanted to ask first about the research, because I'm not in that area, but I'm in human neuroscience, that since the enteric nervous system is involved in communicating, in communication, then do you think at various points from embryo development that you can communicate with those cells, and perhaps through the genetic transcription factors make cells that wouldn't normally be enteric nervous system cells, stimulate those cells to become enteric nervous system cells?
Yeah, so you're touching on a really important point here, and I was a little rushed at the end. The implications of this work, one of the main, the NIH fundable reasons why we're doing the work is we're discovering some of these new signatures that would help us to understand how does that sensory cell develop versus this cell. Absolutely, it could be used in regenerative medicine or reprogramming experiments. And in fact, it already is in different contexts. So there are some labs that are using organoid or enteroid research to use some of this information that we already know from other earlier studies to try to program either neural crest stem cells or other neural progenitor cells into distinct fates. So yeah, that's a very good question, and it's totally yes. The answer is yes. We can probably do that.
Thank you. And in terms of your story, I just found it so inspiring at every point. And I'm wondering, what was your personal motivation to keep going when things were hard? I guess what helped you in terms of the passion for science? And then you mentioned the people, but were there other things that helped you when you might have felt like giving up?
Right, yeah, so in the beginning, I mentioned that it was very difficult, and I ended up finding lab research. Just that was some soul searching and realizing that I really loved science and wanted to stay in it. But as I went through my journey, just really getting reassurance from my community, like those around me, that what I was doing was important and that I was making progress, and that just positive reinforcement, but also critical feedback. So if I was doing something stupid or I wasn't making sense, or like I would get constructive feedback as well, and take it and work with it. So I guess just being very, very proactive. I really, really, really was in love with the science. That was a huge motivation for me. And my family didn't understand what I was doing, but they encouraged me every time that I would tell them about something that happened, bad or good. They would be on the sidelines encouraging me, and that I was the first in my family to even remotely be doing anything like this. I think there was a huge swath of my family who thought that I was in med school the whole time or something like that. Like, that's a really general answer. My mentor along the way in grad school was phenomenal, and he was beyond supportive and very understanding and hard. He knew when to be hard on me when I needed it, but he also knew when to back away and give me space, which was pivotal to my growth as a scientist.
Thank you so much, Dr. Uribe. I can relate to that, to not having anyone in your family understand. I'm also the first in my family to go into science.
Oh, welcome.
So it's been very new for me as well. Thank you so much.
That's very good. Welcome. Hopefully everything works out.
Are there any other questions?
Hi, Dr. Uribe. My name's Ryan. I'm a second-year neuroscience student at UTMB.
Hello.
So I was just interested in, so you talked a bit about the neural tube in the beginning.
Mhm.
And given the importance of like the neural tube closure, what do you think might happen if there are defects like due to like lack of folic acid in the development of this enteric nervous system migration pattern and everything?
Right. Yeah. So you're asking a very interesting question. So we do know from decades of research that if you have folic acid deficiency, that there will be a lot of birth defects, neural tube closure defects. It's like a spectrum of congenital diseases in the vertebrate embryo. But what role that would play on downstream like neural crest derivatives? I think people are just barely starting to scratch the surface on that. So I have some colleagues from the Bronner lab who are interested in looking at the role of folic acid and if it's playing a real true role in neural crest migration, delamination, as well as subsequent differentiation of the derivatives. And I can't say much other than that there is some importance there. I can't give specifics on it because it's unpublished work. We haven't done anything specifically with the enteric nervous system and folic acid, but I wouldn't be surprised if there was something wrong in a folic acid deficient embryo, zebrafish embryo, or if on the other end of the spectrum, if there was too much folic acid. Those are open areas that and I'm sorry if there's somebody here who actually knows of this, like not that I know of, I don't know of any large studies that have really looked at that. It's an open area.
Got you. Thank you so much.
Mm-hmm. Let me see if there's any in the chat. Okay.
Anybody else feel free to shout out.
So yeah, I'll just like say another comment, like good luck to all of you. I don't know many of you, but if you're graduate students or postdocs, I hope you heard some of what I said. Please don't be afraid to, it's hard with the pandemic, but hopefully when we get back to normal, I feel that you can get together with each other and talk to faculty try to build some type of community. That helps out a lot. That's my huge, if you don't remember anything else from my talk, I hope you remember that.
Dr. Uribe, if I could ask another question.
Oh, you have one more? Okay.
I was so amazed by, but not surprised by how you were able to recruit such a great team while starting as faculty. I mean, given the person you are that I've gathered from your talk, but I was wondering if there was anything that helped you to find the right people and I guess to generally start off on a good foot with starting your new lab and getting all the resources that you need.
Right, yeah, so I had a very supportive department, and one of the retiring faculty members, so one of the faculty members was retiring at the same time that I was coming in. And so he had a fantastic lab manager named Eileen, who basically transitioned to help me. And she helped me to basically get the lab physically set up really fast, like within a semester. And her hard work and tenacity, I would not have been able to get set up nearly as fast without her help. So from the departmental perspective, they were very, very good there. Along with Eileen, that first group of graduate students, they were just very interested in developmental biology, that bunch, and they were interested in cellular biology. And so when I was describing my science, they wanted to rotate. And so they were working on various different projects. And so I think they just really ended up coming in and liking zebrafish and working on the neural crest projects. So I think it was kind of like, I don't know what else to say other than I think you have to make sure that the students are interested in the work and that they like me. Sometimes they might not want somebody. So you all know, you're graduate students, you've gone through rotations basically. You know how that goes from a graduate student perspective. But I think it was just the students in my lab, those who joined the lab are really truly interested in developmental biology or stem cell biology. And I guess they saw that I was going to be a good mentor, something in me. I don't know what they saw. But yeah, that's like a big hand waving question answer.
No, I'm glad everything just works out so well and such a great team gravitated toward you and you're all a great team together.
Well, thank you.
Well, if there are no other questions, I want to thank you again for giving such a great talk and really a good, inspiring story. I know that I'm lucky to be in the same department as you and get to interact with you and some of your graduate students on a daily basis. So thank you again for giving a great talk.
All right, thank you, everybody. I really had fun chatting with you this afternoon. Now, have a good weekend. All right, bye-bye.